All right, so let's get started. Welcome to the University of Connecticut Brain Imaging Research Center, or BERC's speaker series. I'm the host, Fumiko Haeft, Director of BERC and Professor of Psychological Sciences, Mathematics, Neuroscience, and Psychiatry. Starting this fall, we have merged the traditional hybrid speaker series that was primarily in person and the speaker tag series. The speaker tag series is a newer series that we started this spring, and I see a lot of familiar faces that has been coming here, so thank you very much for being a repeated customer. And it's unique in that the speaker tags the next speaker. In Damien's cases, we're restarting this fall, so I had the liberty of nominating him. Since April, we've had Daniel Ansari, Lucina Udin, Nathan Sprung, Danny Bassett, Daphne Shohami, and Roshan Kouls, all fantastic cognitive and computational neuroscientists. With the class started, we're now merging the two, and we'll have a bit more of a complex schedule, some that are tagged and some that are not, but both on Tuesdays, with some in the morning for East Coast time and some in the afternoon. So please join our listserver if you want to stay on our schedule and be notified, or check out our Twitter. So we've had about 100 to 200 people each session from over 100 institutions participating each time. And as you can see in this slide, you can view previous recordings such as Tor Wager's, Jim Haxby's, Kim Noble, and others on our website. And as I said, you can also sign up and get updates on our talk series by emailing Lizzie that you see on this slide. Let me take a break here and try to admit more people who are coming in. So some housekeeping items: you will be muted, and clapping, we will unmute. I usually try to unmute to allow everyone to clap because I think it's always nice, even though I fail often and there's often something that goes wrong. But if you have questions, please enter into the chat box. If it's urgent, Damien is happy to answer, but please put urgent at the beginning of the chat box because I am the only person moderating that today. But we will take most questions at the end. So let's get started. Today it is our great pleasure to introduce Professor Damien Fair. Damien is a computational neuroscientist and a newly minted founding co-director of the Masonic Institute for the Developing Brain and professor in pediatrics and the Institute of Child Development at University of Minnesota. He has a physician assistant degree from Yale and a PhD in neuroscience from Washington University School of Medicine, where he worked with Brad Schilliger and Steve Peterson, some of our favorite cognitive neuroscientists. He did his postdoc and assistant associate professorship at Oregon Health and Science University, and he has just moved to University of Minnesota in his hometown. He has quite a remarkable and fast-track research history, as many of you know. I'm going to take a break here and admit more people. So Damien's research focuses on mechanisms and principles that underlie child and adolescent brain development. The majority of his work uses functional MRI and resting state functional connectivity to assess typical and atypical populations, such as ADHD and autism, though it's much broader than this, as many of you know. For example, he founded a company and is chief scientific officer at Neuroimaging. He is currently a PI on 10 NIH grants, and has also been funded by Gates Foundation, McDonald Foundation, MacArthur Foundation, where his work extends into emotional algorithms, impact of childhood trauma, and opioid exposure on brain development, cross-species imaging, and on the ABCD project that we all love. He has published more than 100 articles in high-impact research journals, including Nature Neuroscience, Neuron, PLOS, Computational Biology, PNAS Science, and Psychological Science. He has received many awards for his work, including the Presidential Early Career Award for Scientists and Engineers, issued by President Barack Obama, and not Trump, and the White House.
Don't delay thing you can do now.
And as we just learned, many of us just learned, he just learned today that he has won the MacArthur Genius Award. So we want to, I will have a time where we can clap for him, but congratulations to Damien. And what I find very importantly, he is a champion for diversity, equity, and inclusion efforts. For example, he was the founding director of the Oregon Health Science University's President's Fellowship for Diversity and Inclusion in Research. I always like to say something personal. I apologize, it's getting a little bit long before we can get to clap and hear his talk. But I think we first met back in 2008, and I heard from Brad Schilliger many years ago, and I confirmed just with Damien today, but At that conference in Berkeley, I was giving a talk on a workshop sort of on machine learning back then. It was many years ago. And he just told me that his whole line of work on machine learning got started then, which I feel very honored to hear that from him. And so with that, I want to unmute everyone to give an opportunity to clap and welcome Damien. So I'm going to unmute everyone. If you have kids and dogs in the background, they are welcome to clap as well.
Hey, congratulations, Damien.
Congratulations. Oh, hey, Bruce.
Really, really thrilling.
Bravo!
Amazing.
Yes, congratulations. So let's get started. And do you want to take over the screen?
Yep, I can do that. Hopefully this will work. All right, can everybody hear? Can everybody hear me?
Sorry, I had to mute you for a little bit, but yes, you're back on.
Okay, let me see if I can get this to go. Just 1 second. It's, for some reason, the buttons aren't working here. Let's try this one more time. Okay, can everybody still hear me and see the screen?
Yep.
Okay, good. All right, so I will get started here. First of all, thank you for that great introduction, and thank you for having me talk about some of our work. And I will tell everybody that and Bea knows this probably more than anybody, that I tend to procrastinate a lot, and getting this talk completely kind of polished, I was procrastinating too, imagining I was going to today, finish off all the pieces and the tweaks that I wanted to do for the talk prior to actually giving the presentation. I didn't really appreciate the deluge of information and good wishes and media reports and everything that was going to come with this new award. So I didn't get a chance, like I wanted to fix all those pieces, but hopefully I'll be able to at least give you the gist of kind of the direction of a lot of the work that we're doing. I always like to start my talks in part by, highlighting a lot of the names. This is probably not even everybody that contribute in some form or fashion to a lot of the work that we do, and things that I'll be talking about today. It really does take an army of brilliant people to conduct, the kinds of work that we do. It's very collaborative. Physicists and computer scientists and biomedical engineers and neuroscientists and clinical psychologists, and it's just a lot of amazing people that kind of come together to make the work go, and it's really very much appreciated about that. And I'll highlight some more people as we go along. There's a few main goals that I have for the talk today. I'll be describing a collection of data for the ABCD study that we call the ABCD BIDS community collection, and some of the data and utilities that are now available. There's a paper that will be coming out on this relatively soon, hopefully before too long. So a little more work to do. Then I'm going to highlight the replication problem in our field, in other fields, and how it really is kind of a turning point for how we are likely to have to do the type of work that we do, going on in the future. Work by Scott Merrick and Brendan Turbo-Clemens and my colleague Nico Dozenbach that I want to highlight. But then, of course, instead of perseverating on that downer, trying to describe a few things that we're doing to deal with it. And one of those is trying to increase the SNR in our data, particularly functional connectivity data, by utilizing information from individualized networks, and then maximizing effects potentially by leveraging large data sets to study smaller samples. Okay, so the ABCD BIDS community collection. All right, so this particular collection and data and analytic utilities were designed to, in essence, supplement the data sharing that's currently going on with the primary ABCD repository on the NDA, but that it's accessible, but also it serves as an accessible platform to actually share derived data of individual origins that otherwise is difficult to do with ABCD data via the NDA. It's also meant to kind of assist investigators in conducting, state-of-the-art kind of statistical analyses using various types of software packages designed to work with the data set. And it provides some other integrative data and analytic utilities to assist with result verification and replication of your data. And at the bottom here, I highlight a few of the information, the docs, and where you can grab some more information on the the collection altogether. I won't be going over everything, but I'll show you a few things now. Now, the data contains these BIDS derivatives that right now, there's derivatives in there from what we call the ABCD BIDS pipelines. There's free surfer outputs, connectivity matrices, things of that nature. We're currently working on uploading and releasing the ABCD data via fMRI prep, their most recent versions, QSI prep, things on individualized networks, and the task fMRI. Hopefully by the end of the year, some of that will be uploaded and prepared for folks. And there's several utilities. We'll go over very briefly some of those here. The ABCD BIDS pipelines, the ARMS, which we call the ABCD Reproducible Match Samples, SEND, which is a sandwich estimator for neuroimaging data to allow proper statistics on this large data set, and some download and upload tools, matrix integration tools, and several other things that you can identify at some of these links and locations below. And these are just highlighting the two some of the repositories that are available where some of this exists. All right, so the ABCD BIDS pipelines, these are in essence based on the human connectome pipelines that you can see in the middle of the screen here. But they're containerized in Docker. They utilize lots of additional tools, kind of the best of much of the tools that we're all familiar with. So it can provide standardized inputs, minimalistic interface. It makes things just really easy to use. So this is just the command line of how you would run the app, and the only thing it really requires is a path to your BIDS inputs and outputs, your free surfer license file, and that's it. Now, there are several options that you can do, but really it's meant to be really minimalistic and easy to use. The other important thing is that it works with ABCD data, legacy non-ABCD data, and with special populations, particularly aging populations where they're different sizes of ventricles and things like that can make some of the data processing more difficult. This allows for really high consistency. All of the data that's in this new data set is in the BIDS format. We put a link up there for folks who are not unfamiliar with BIDS format, but really standardized way of organizing your data and labeling your data so that it can be utilized with any type of BIDS app really easily, and makes it really easy for sharing. The derivatives are simply the outputs of a given application. The total standards for these are not complete, but we've been following the current standards and been working with the team there to develop new ones where needed. And importantly, the way that this new collection is set up, that the downloads from the NDA for the ABCD data can be type specific. So it assists with facilitating usage and reducing costs. So you don't necessarily need to download everything to get one little file if that's all you want. And again, there's many tools to help with that process. This is not showing up too well, but there are, as part of the ABCD BIDS pipelines. There's lots of QC information, including things like these gray plots, so you can make sure that your processing steps are really reducing or getting rid of all the noise in your data. There's other HTML and other tools that let you scroll through your data, looking at the surface, how well the surfaces were constructed. If there's any problems with the registrations and things of that nature. We call it the executive summary for the QC. And just there's all the data are now processed and available for download in this particular collection on the NDA. Remember, there's 11,000 over almost 12,000 subject scans, 11,000 available on the fast track on the NDA. And as long as they have a T1, they're able to go a good T1 that have passed QC, that be able to go through this pipeline. So that's upwards of 10,600 subjects or so that have gone through this process. So the other, one of the other tools that we think is important to kind of let people know is when they're using these data, is like what we've been calling SEND, of the sandwich estimator for neuroimaging. So the brain-wide association studies, whether you're doing connectivity, cortical thickness, or anything relating the brain to some complex behavior, most often has to take into account some type of covariates, right, that may impact your analysis, right? The ABCD study is complicated because there's a lot of covariates, that are normally independent, but that in this case are nested. So what that means is that, from all these 21 sites, just one example of many of these, right? You have MRI data that are acquired across 3 different scanners, right? So you have site and platform effects that are likely to impact the measured data. But because not all the dependent measures are overlapping, so not every site has a different scanner, right? The covariance structure of the data is then nested, okay? And that can cause some difficulties. So typically this nested covariance structure can be accounted for with what we usually use, like these mixed effects models, linear mixed effects models. But in imaging data, when you're doing voxel-wise or gray ordinate-wise or connectivity structure-wise data sets, there's tens of thousands of elements, right? And then there's thousands of subjects with repeated measures. And that means that after you start really digging into it, you have billions and billions of data points that you're trying to do these statistics over, right? So then the other aspect of this is that as you get more and more covariance structure, right, or more and more covariates, kind of properly adjusting for statistical significance can be difficult with imaging data. So what folks usually refer to this now, they do some type of permutation testing or something of this nature. Well, as you add more and more covariates, and how you have to do the permutation testing becomes less reliable as the number of tested variables in essence increases. So the marginal model approach, which came out of Tom Nichols' lab, was developed, and we made software packages to make it really easy to use with ABCD data that we call SEND. It's really kind of simple. Well, my cursor's not working, but in A here you have, you can very easily construct a model. You can do in this case, on either surface or gray ordinate data or matrix data or volume data. You can do something if you know something about the network structure of the data. You can do certain types of enrichment analyses to look at network types of effects. And then because the permutation testing, in this case it's called the wild bootstrap, and this is what's in B, is done on the residual error, that it makes for doing the proper statistical testing allowable, because you're able to do, essentially do the bootstrapping on the residuals instead of doing it on the actual data. It's called the wild bootstrap, and that allows to make sure that your tests are truly significant. Now, probably the most important thing as we start to get really complex analyses is that the performance comparison compared to your traditional mixed models relative to the marginal model is night and day. It's much, much, much faster, and allows for studies that don't have monstrous computes to be able to be conducted even with these very large data sets. So another data utility is what we're calling arms or ABCD reproducible matched samples. Right now, and this will be a theme of the talk, is we're at a reproducibility crisis, I would say, in our field, where a lot of the findings that we have been conducting in more recent years or over the past 15 years are not highly reproducible. In this particular paper, they're relating this reproducibility problem to differences in the methods or procedures of how people are conducting things, but there's other reasons why that is too, and we'll go over that more. But one thing that we think is important and is allowable, with having such a large standardized data set like ABCD, is that you can split up the samples into really true test and train or reproducible samples. So here what we've done is we've taken the entire ABCD data set, we've split it up into, in essence, 3 groups. Group 1 and Group 2 are 2 large, nearly perfectly matched groups on lots of demographics, ethnic backgrounds, family structure, and things of that nature. And then a third group that is also matched, but is much smaller, but used for model building and some other types of testing. Okay, you can kind of see the means in here, but these are very highly matched groups, and the data are very tight. That allows, again, and I'll show you how I'll give you an example of how you can use it to make sure that your data your findings are actually replicable independent sample. Okay. So what we're looking at here now is simply taking those two, those two, what we call ABCD1 and ABCD2 groups. We're averaging the connectivity matrices of these 2 independent samples in A, and what you can see is that they're highly related. This is one of the things that weren't able to fix, but the correlation, the point estimates here are almost nearly identical. The correlation between these two measurements are over 0.99. B is simply looking at the variance. Again, the point estimates are extremely tight, meaning they're highly reproducible. And the same goes with these 2 groups looking at measures of, for example, cortical thickness, which you can see on the right. All right, and so just to give you one simple example that kind of pulls us into the next part of the talk is based on this paper that came out on the ABCD study by Wes Thompson back a couple of years ago, which just in essence identified the principal components model to break down a lot of the cognitive measures into these 3 identified traits: general ability, executive function, and learning and memory, or working memory. And what we've done is we've kind of run these models independently in these 2 groups, which replicated this initial data set, but it also can give us a good exemplar of what we might expect what we might expect with regard to reproducibility when we're looking at data in these 2 independent samples. So here, what I'm showing is the brain-wide association of general ability and the connectivity between the connectivity measures and general ability from that PC for both the first ABCD1 and ABCD2. Now that 0.67, that's actually an R squared. There does seem to be a lot of relatively good reproducibility with regard to how these measures relate to, if you're using all the data, relate to general ability. But as you can see in D and then I'll show you again in E, right, is to get that level of reliability, it appears that you need to have a fairly large sample, right? So by taking subsets of one group and comparing to the gold standard of another, so taking subsets of ABCD1 and compare it to the gold standard of all the data in ABCD2, to reach that level of reliability, you need lots and lots of data. And that's kind of what's shown in D, but it also shows the vast variability that you'll get with samples as low as 200, 300, right, 100 and lower. Lots of variability in what you might expect also has low reliability. Okay. Importantly, if that's what data where we were used at least 10 minutes of good, non-movement free data from the ABCD subjects. If you move that data down to only 5 minutes of data, that reliability drops quite a bit, and it gets even worse, okay? So in this case, that you need, to get the 80% reliability, which is that 80% of the peak, which is that dotted line right there, in both D and E, you need in excess of almost 500 subjects if you only have 5 minutes of good data. And so that begins to make us a little bit scared about what we should expect and how our data and how, as far as reproducibility in general in our data. And the other aspect of this that I think is important for folks to note is that, so A and B is in essence what we just looked at, and C, D, and E and F are now looking at the other principal components of working memory in E and F and learning ability, and C and D is executive functions. Now these measures, the outcome measure, in this case the PC, was even less reliable than the general ability, and so the reliability of these measures is even lower than the others. And so it just highlights of how you need to really maximize your reliability in your data. You need lots and lots and lots of subjects. Okay, so the gist of this first little part of the talk here is that the data are looking good and are reliable in ABCD. The ABCC, the NDA Collection 3165, provides an additional and growing resource for the community to conduct analyses and actually share their data with the ABCD data. But despite all the efforts to maximize the signal, data harmonization, due to the remaining noise and likely what we call sampling variability, which I'll go over in a second, our questions using ABCD or really any other study are likely going to require much larger sample sizes or group membership than we had previously imagined. And that gets us to this next part of the talk, the replication problem in our field. And now I think we're kind of at a place where we're at a crossroads. So I have had the pleasure to work with some amazing students, Scott Merrick and Brendan Turbo-Clemens. I've had a long time been working with Nico Dozenbach, where these guys, did a really deep analysis into this replicability problem and why we might be seeing it. This paper is now out on bioRxiv. By the way, both of those students come from the Luna Lab, which is telling us that we have a really great mentor over there in Pittsburgh. But what I'm going to show you is, in fact, these are a lot of these slides, in essence, come from some of Scott's talks. But I'll show you some of the difficulties and some of the things that we really have to worry about for a lot of our analyses in cognitive neuroscience. Okay, so the first question here that's asked is, so what are the effect sizes? What are the expected effect sizes of our brain behavior correlations? What should we be expecting that we should see? Okay. Now, for this study, it was important to reduce the sample from, the approximately 10,000 subjects down to the cleanest subjects, which gets us down to about 4,000 people. Okay. And then we're hoping that this can give us a more precise estimate of the effect size of these brain behavior correlations. And through resampling, the same types of resampling techniques I talked about before, examine the effect of sample size on actual reproducibility. And I'll just note that even though we did the first analysis on the first 4,000, it was replicated again on the full data set as well. All right, so there's lots of different types of measurements in brain MRI. We can look at data in terms of vertices, in terms of specified ROIs, in terms of networks, edges, in terms of connectivity. It can be functional and it can be structural. So we looked across all those different types of measures, and then also looked across 41 different measures, typical measures that are measured in ABCD, including demographics, measures in cognition and mental health, to see if we can get a good sample of what we should expect with regard to effect sizes if we compare across all this stuff. Okay. Now here's the it's really the crux of the paper. is that across all that stuff, right, you're either using cortical thickness, which is on the left, or function, in this case, functional connectivity, which is on the right. The largest univariate effect size across any of those measures, anything we possibly would do, was 0.16, okay, an R of 0.16. The median of the top 1% was 0.06. I'm sorry, was the top 1% was 0.06, and the just 1 second here. Oh, I can't get to it. And anyway, the point here is that the effect sizes across all these measures are very small. Okay, they're much tinier than we probably have seen in the literature. You know, likely for various reasons, which I'll try to explain right now. Okay, so does the reliance on these typical neuroimaging sample sizes, does that provide us with some explanation of why we might have replication failures in these brain-wide association studies? And if so, why? Okay, so the first concept to make sure that we're understanding here is that of sampling variability. And that is how much an effect size estimate varies between different samples of the population, okay? This is extremely, extremely important concept. Okay, so just to give you an example, this is actually data from ABCD, okay? Where if we take from all the subjects, right, we know that height, for example, is related to, particularly at this age, is related to age. Not perfectly, but we know it's related. Okay, so if I take one sample from all of these 4,000 participants or 10,000 participants, I might find a small sample of 25 subjects that have an R of, you know, correlation of height and age at 0.85, right? But I might find another sample of those 25 subjects where that correlation is in essence 0, okay, which would be probably not a true estimate of what the relationship is of height and age. Now, if I do that over and over and over again across all the participants in ABCD, right? On the left side, it's hard to see, and I can't point, unfortunately, but you can see kind of a cloud, the distribution of what you might expect. And what you'll see is you'll get some distributions of samples of 25 participants that are nearly close to 1, where the relationship between height and age is perfect. And you'll get other populations that you'll pull where it actually be negative, where the answer would show that there's a negative relationship between height and age, okay? Now, the more and more sample that you get, the sampling variability drops, and you get closer and closer to the true answer, the true effect, which is that purple line in the middle of the relationship between height and age. Now, this is another example. It's just showing random data, okay, where you know, and again, there's no noise in these data. They're perfectly, perfectly fit, so to speak. And this is random data. Of course, the true relationship between the two effect size should be 0 between perfectly random data. And after you get a large sample size, you can easily see that here. But you also see at the smaller sample size, you can easily be fooled, where you see a relationship that is relatively high. Okay, this is in essence sampling variability. And if our sample sizes are not high, it's easy to get fooled that you might have an effect when there really is not. Okay, now the only way to deal with sampling variability is really to get a large enough sample size that handles the true effect. Okay, now this is different from kind of other systematic sources of error, and you might, you know, in the literature you see a lot of effect sizes that are much higher than would maybe be expected of showing this statement. That's partly likely because of publication bias, where only positive things are published. It likely, if everybody published all the results and all the negative results and everything, we'd probably be, in these meta-analyses would probably come down to the true effect size. Unfortunately, that doesn't happen. There's other things like p-hacking and head motion, other systematic sources of error that if we can correct, we may not need as big as samples as, which I'll show you later. But at least the way that things are currently oftentimes done, we need very large samples. So what do small effect sizes and sampling variability mean for small sample studies? So this is actually just this is 1,000 bootstrap samples at these various sample sizes, okay? that we're showing for structural in E and functional in F. And at the lowest end, in n equals 25, you can actually get what we call sign errors, effects that are completely opposite of what's real. And so again, it just kind of highlights that it's easy to get fooled if their samples aren't large enough to measure the true effects. And so here we're showing that the observed effect sizes suggest that consortium level data may be required to get a truly reproducible univariate braid and phenotype association, upwards of 2,000 subjects. So one of the early criticisms of the work is asking whether this is very specific to multi-site, adolescent ABCD study. Well, in essence, it's likely that that's not the case. So this is data from the Human Connectome project. But the Human Connectome project doesn't have as large a sample as ABCD, but the sampling variability using the same tools, like this is the NIH toolbox, in adults on one scanner are in essence identical. They look quite similar. So it's likely not a case of increased noise simply because the data have been collected at multiple sites. And even if you look at the same kind of measurement within a site, within given site, that in the ABCD, you see that the trends are nearly identical. So it's likely not the case. It's simply because it's a multi-site study. So do multivariate. So in case instead of looking at just one connection or one vertex at a time, do multivariate methods improve replicability? The answer to that is actually yes, they do, but maybe not as much as we would like. So on the left is for cortical thickness. On the right is for functional connectivity, comparing cognitive ability, psychopathology, aspects of the NIH toolbox. Where you have a discovery and replication sample. This is again using that arms split up that we talked about earlier. And here, in the discovery sample, doing cross-validation or whatnot with the very small sample sizes, it is still easy to get fooled, and you still need relatively large sample sizes to get the true results. Not surprisingly, in a completely held out sample, the relationship drops, but there's still some relationship. The point is that you need large samples to get to even the true prediction effects for these multivariate analysis, even though it's a little bit better. And it also seems to be quite a bit better for resting state connectivity as compared to cortical thickness, at least in this example. Okay, so does this provide a parsimonious explanation for replication failures in BWAS? The answer is yes. The coupling of these small effect sizes with relatively large sampling variability produces statistical errors that undermines reproducibility. And if you want to really dig into the theory and the essence of what we're talking about here, there's a nice paper at bioRxiv that was written by Anthony Dick and Wes Thompson that more recently came out that goes over some of these issues in really good detail. It's a very nice read. Okay, so and some of my other talks have ended there, and it's like a total downer, right? Because it's like so depressing, that that in order to really do this right, we need large samples. Okay. But we have a few ideas and a few things that we're playing with. A lot of the stuff is hot off the presses. It's not out there yet. Hopefully that you guys won't go out there and scoop me before we can get it out there. But I wanted to show the group some of the things that we're doing to, in such a squeeze out some of the error that's unrelated to sampling variability, and see if we can improve some of these effects. Okay, so there's other things that we can do. Of course, a lot of aspects of standardization. To pre-registration, so people can publish their data, whether it's positive or not. Code sharing, things like that will also make it easier for data aggregation to increase. And those are all things that we absolutely should do as a community to assist. I'm going to show you a few other things too. All right, and again, so what I'm talking about here is not changing the sampling variability, but on the bottom right, I talk about these other systematic error sources of error. If we can squeeze out some of those other pieces of error, then maybe we can help ourselves with regard to the samples that are required. Okay, so to start this, I'm just going to remind everybody a part of how I, in essence, started my whole career in this space, and that's when we came across, trying to use graph theory, right, to characterize brain structure and organization. Okay, now simply, graph theory in this case is the study of nodes and nodes and edges that form networks. In our cases, in this case, the nodes are our gray ordinates, and the edges there are there's that connectivity between them. Now we've been using what's called community detection to identify in a data-free way, in a data-driven way, where these networks exist. Starting, it seems like forever ago, way back in 2009, we used community detection to identify specific networks. That area of work has flourished and is really taking off in ways that I never could have imagined. But now we have a pretty good sense that there seems to be a relatively small number of large-scale systems, at least from a 10,000-foot view, that exist in the brain. They're highly consistent, from at least early childhood through adulthood, and are highly replicable as long as the data are strong, and certainly if the samples are large enough. Now, most of this work has been done up to date, has been done on the average brain, right? So where we have we take a bunch of participants, we average their brains, and we identify where the communities lie. That's primarily most of what we know about these systems. But in recent years, we've been found out that nobody's average, right? You have the above average brain imaging research center at UConn, and you have the below ones, right, that exist over at Yale. Now, I'm joking. I can give Todd a little flack here. I actually did my very first, as a physician assistant, my very first fMRI study was that residence imaging center that Todd ran down at.
Todd's not on today, so.
Okay, well, good. Good. I mean, the point is that nobody's the same, right? People are very different, even though most of these regions and things that we're looking at are all based on average brains. Okay. Now, the Poldrum, Russ Poldrak, who's a crazy person, right, decided to scan himself for 10 months, twice a week, to just collect an enormous amount of data on his brain. Tim Lauman and my old mentor, Steve Peterson, did an amazing paper published in Neuron that's really described the network characterization of Russ's brain. And it was the first really we wouldn't say the first to identify these individual brains and these functional fingerprints, some of that stuff that we had been doing for a long time. But it really kind of showed that the network topology and topography is very different, from person to person, even though the general structure all exists. That paper was followed up by this Midnight ScanCom paper by my colleague and friend Nico Dozenbach, which did in essence the same thing, but in 10 participants to show how variable it is from participant to participant to participant. It's about 5 hours of data in every subject. And I'll go back to ways that we're using this data shortly. But really, as you can kind of you can see here with these arrows, there's just this very unique, and it's highly replicable, but there's these very unique ways that the individual brain is organized that you just can't see when you're looking at the average. And due to time, I'm going to skip over this. You're just going to have to believe me that it matches up with tasks and myelin mapping and cytoarchitectonics and things like that. But it's really, this individual network structure is really real. The problem with a lot of this work, though, unfortunately, is that you just need an enormous amount of data to make it go, right? So in the case of the Poldrak paper, they did this very similar type of analysis that I talked about earlier. You take half of Russ's 7 hours of Russ's data, you make it the gold standard. You take subsets of his data at different, different lengths, different amounts of data, and you see, well, how much data do you need to reach the gold standard? And in this case, what we identified, or what was identified in this paper by Tim, was that you need to reach approximately almost 95% of the max. You need about 40 minutes of data to kind of reach complete asymptote. You need almost 100 minutes of data. So that is a lot of data to be able to to get a reliable the perfectly within subject kind of matrix. So in ABCD, of course, I was proposing that, yeah, let's do it. Let's do 40 minutes to an hour worth of data at every single kid, 10,000 kids for the next 10 years. But I didn't win that battle. But we were able to convince everybody that we needed at least 20 minutes of data. Now, on that blue curve, the bottom point there is 10 minutes of good data. And so as soon as you reach about between 10 to 12 and a half minutes of data, you can get to about 85% of the max, or an R of 85% of the max, and we felt that was about as much as we could get with ABCD as far as reliability within subject. All right, now one of the things, in fact, there's a new paper that just came out it's in bioRxiv on this, is that over the years, we've been searching for ways to be able to get similar types of reliability with regards to network structure in individuals, but that didn't require as much data. One of these methods is called template matching. There's a version of this that started off with a paper by Evan Gordon in 2017. This is work that was primarily conducted in the Peterson lab, but we've been tweaking with, playing with it over the course of the last couple of years as well. And the idea is it's more of a supervised way of identifying these networks, where you might start off with a network system like this based on the group average. You can identify a mask or a seed essentially for every given network. That seed can be used to generate a map of the connectivity essentially of that seed of each one of those networks. And then when you have an individual, you can take every single gray ordinate, every single voxel, you generate a map and you compare it to each one of those templates. In essence, this is a winner take all system. So the template that it looks like most close to you, it's the winner. And then that voxel is assigned that network. You do that for every network, every voxel, and you can start pulling out networks that look remarkably like the ones that we described, that were described earlier in the Lauman and Gordon papers that use 40 hours of worth of data. So there's two goals here that we'll go over quick. Is that the first goal is we want to validate this template matching such that it can mimic the gold standards of what we've been doing with community detection. And then ask the question, determine the amount of usable data necessary to get an accurate, individualized, personalized system. Again, it's a little blurry, unfortunately, but what we've done is we've taken the Midnight Scan Club data, again, has 4 to 5 hours worth of data. We split it into 2 halves. And then we can run our new template matching, or we can run community detection on each one of those halves of data. And from there, you can then, with mutual information, you can compare within subject versus others. You can compare how well the specifics of the systems matched. Okay, and again, that's within subject and then between subject. And then you can do that for community detection method versus community detection. You can do it for community detection versus template matching, and so on and so forth. Okay, or template matching versus template matching. All right. So the first part here, I'm going to show you, well, I'm just going to show you an example of a single subject from the Midnight Scan Club data of using community detection, and the other on the right using template matching. And you could see that, at least at first blush, it looks like a lot of the interesting variants that you see within this individual are kind of replicated across the two different methods. Okay. But now I'm going to show you the distributions across everybody. So this is what the distributions of mutual information of self versus other using community detection look like. You can see there's lots of separation, not surprisingly, between self versus other. this is, is that my mic? Oh, just 1 second. My Mac is going dead here. Oh, it should be good now. Okay. All right, can everybody still hear me?
Yes.
Okay, good. So sorry about that. It looked like my computer wasn't plugged in all the way. In any case, so on the right and now on the right side, it's looking at community detection versus template matching. What you can see is that the distribution, the separation of these two, the distributions are quite similar, suggesting again that they're both can kind of separate the groups quite well, and that they're showing similar kind of findings, even if it's not perfect. Okay. And then the next thing we can do then is say, okay, well, within template matching, let's do our same thing. Let's take a gold standard of 1/2 of the data within subject, and then we'll compare it to 10 minutes of the second half, 20 minutes of the second half, 30 minutes of the second half, and so on and so forth. So we did that, and what you can see is that and this figure is not totally complete, but what you can see is that you start to reach an asymptote, even though it's again not perfect, even as low as 10 minutes of data instead of 40. Okay, so this is giving us some confidence that the network space, the network organization topography within subject is good with template matching, and we can be done with much less data. Now this is just a quick, GIF of 200 ABCD subjects. And you're just looking at the individual network topology and the variation across all the participants. You can see here that it's even though the general structure is maintained, there's lots of variability. And we think that we can use that. Okay, so in this sense, and here what we're doing is we're saying, okay, we know that there's the general organization is tight, but we know that there's likely some voxels or some gray ordinates in the brain that are highly consistent across that network. So here what we're doing is creating this precision or probabilistic functional connectivity atlas, okay, where it's just a probability of a given gray ordinate or voxel exists within a person, across the population. So the bright is very high probability, and dark or nothing is very low probability that is in a given network. Do that for all the networks, and now you have this probability map. The nice thing about these, one thing about these probability maps is you can see when you compare an A to D, is that if we take our ARMS data sets and we split the sample and we generate one versus the other, if the probability maps are very highly replicable, almost perfect, down to the specific gray ordinate. We also know that if you run, at least in a small sample where we're able to run community detection, because they had enough data, that the relationship between the gold standard community detection with this template matching in the ABCD data set is also quite high, with a correlation of and again, this is group 1 versus group 2 of 0.95. Again, very tight. And that goes for both this cortex and the subcortical structures, which you can see on the bottom right. This is just for one network, but you have to trust me that the other networks were quite similar. Okay, so last part here that that we think is important is that this gives us an opportunity now to, instead of defining our regions or our parcellations that we have across these different networks based on group average data, like we've done in Gordon parcellation, but many others, we can define those parcellations based on the probability actually that a given voxel will be consistent across the population. On the left side, we essentially cut this off at an 80% probability. So now we have regions across all the networks that that every single gray ordinate there is at least 80% of the sample has that network for that gray ordinate. Okay, now these types of parcellations might not be used for every type of question. For some of our predictive analyses, they probably will help. So on the right side, you can see the matrices, and just by eye, you can see the within versus between network precision. The correlation values are quite a bit higher within network. Not surprisingly, if you use this 80% probability parcellation instead of the traditional one that we use below. And this decrease in noise, we think, is going to help with our predictions. I just, you know, if you want to quantify this, we did this over here. This is really hard to see, but the dashed lines are, you know, within subject connectivity for the probabilistic maps, and the dark is the same for the Gordon atlas. This is just for one group. These again are highly replicable, but what you can see if you look across is that the connectivity is just higher, at least within network, by quite a bit, using these probabilistic maps versus if you use traditional kind of parcellations. Now, remember this slide where I kind of was starting highlighting this brain behavior correlation stuff, right? And showing, you know, that you need these really, really large samples, right? And that was using the Gordon atlas. Well, now we can actually compare do that same comparison using the probabilistic parcellations instead of this one that are based on the average. But that's what this slide is showing right here. The blue line, this is one thing that I was not able to finish. The blue line is the probabilistic parcellations. The orange line is the Gordon, is what we saw from before with the Gordon parcellation. And the top left is general ability. The bottom is executive function. The top right is learning and memory. And what you can see from all of these is that the reliability goes up, and by quite a bit, if you're using these probabilistic regions instead of the traditional Gordon regions. And in fact, even though in general ability the fix is small, so to reach the same level of reliability, you need about an extra 180 subjects or so. If you're only using the Gordon parcellation, but the difference in the other comparisons is quite stark. So the same, you can reach the same reliability with these new parcellations with upwards of 300 to 300 to 400 subjects fewer to reach the same level of reliability. Okay, so we think that this approach may again begin to assist us in helping us handle some of the sample size issues that I think we're going to need to deal with in the literature. All right, so, oops, okay. The last part here, the last part of this piece here is I'm just gonna highlight some other ways that we're looking at the data to assist with this problem. One is looking at what we're calling integration zones. So it's a way of looking at, instead of thinking of the networks in the brain as unitary, meaning every node has to be in a separate network separately, we can look at them using, thinking that these networks are likely to overlap. Well, we have a method for that now, and again, this is hopefully be coming out before too long, using the version of the template matching where we can look at the distributions of how every voxel relates to all these different templates. We can set a threshold based on, you see that in the bottom right, you see these bivariate distributions. Instead of making a winner take all, we have these algorithms that separate these two distributions and then allow all the voxels within the right side distribution, in essence, to be part of the network. And that allows every network to be estimated where it exists independently, and then allows for overlapping systems. Now here's just a picture of all these all the different systems that we exist. This is just within one individual. I'm just pulling out one. This is the ventral attention network. The blue highlights there highlight what you typically see with the ventral attention networks, but there's lots of other brain systems that are involved in that major distribution that I showed you just before. Now, what's cool is that if you do this, and this is just in one subject, you look across everybody, and what you can see is that in some parts of the brain, there are extremely high areas of overlap. These high integration zones where upwards of 8 different systems can come together. Some of these pictures look very, look familiar to most folks, particularly those high areas of high integration. Default areas, posterior cingulate, rostral cortex, things of that nature, medial prefrontal cortex. But we think these integration zones may also be important for how we examine our data. So this is what this data looked like across half the sample, about 4,000 participants, okay? And what I think is most fascinating, you can see, again, some of the areas that pop out really nicely. I think what's more fascinating is if you look at these integration zones as a function of subcortical structures, you can see some very cool things where you see these very tight integrative zones in the thalamus, in the caudate, putamen, the hippocampus, where you see there's lots of integration between systems, and various areas of the cerebellum. So we're making this new probabilistic atlas that includes these integration zones as well. And just like some of the other measures, these maps are highly reproducible, almost to perfection. All right, so to close this part here, and then we have one more section left, is these traditional approaches to these individual networks might be difficult outside of subjects with maximum amount of data, of movement-free data. How this identification is possible with ABC data using alternative approaches like template matching. All ABCD subjects of sufficient data have personalized networks run now, and which will be released via this ABCC on the NDA, as well as these new overlapping network analyses, which also show promise with these data as well. All right, so the last part here is, again, is like another way to kind of potentially do small studies and maximizing effects, leveraging large studies like the ABCD and ABCC. Now, this idea actually comes from Anders Dale's lab, where they have been treating the brain in their network, in their cortical thickness analyses, very much like you would do in genetics. The same problem that we talked about today, happened in genetics, 10 to 15 years ago, and they figured out ways to deal with this. And one way to deal with it is kind of combining lots of small effects into into a polygenic risk score. In this case, it's a polyvertex risk score. Okay, so most of us have heard of how this works, right? You have the baseline big data, you can identify the betas, then you have a target sample that may be a lot smaller that you can use these combined effects to generate larger effects. So you can do these studies in smaller samples. There's lots of processing QC. You do some LD adjustment for correlation. You can set some P threshold. This is how all generating the betas. And then you test it. Again, this is on the bottom. You can test it using generate the PRSs and test it using independent out of sample sample. Okay. Well, the process here is very similar, and I'm not going to go into this again in very detail. This is part of my wish. I was hoping to get this part of the talk cleaned up before now, but I was unable. So I'll just give you the gist of how we think this works. Again, this will be out before too long. You'll be able to see it. But we have these two different data sets in ARMS, right, where we can generate these. We can see how replicable our brain behavior relationships are. And then we also have this another data set. It's longitudinal data set of about 400 kids with ADHD and controls that have been followed over the course of 10 years. So every subject has 1 to 5 time points. Okay, a completely independent sample, nothing to do with ABCD. We run our analyses, and one of the things that we can do is we can test to see how good this works with our new parcellated region sets, and also our traditional ones like the Gordon et al. Do the processing. We can do same types of adjustments based on correlation structure in the data. We can apply to generate the betas. We can use our SEND, our sandwich estimator, to be able to generate good betas that control for the nested variables, genetic, or the sex and things of that nature. Generate the PRSs, and then we can test in this completely independent sample. So to make a long story short, I'm just going to show you. First is that we did this on ADHD symptom scores in ABCD, and this was work that's done by Joel Nigg and Michaela Cordova, who's now a student at UCSD. And so we made symptom scores that are based on the case ads and the CBCL. Hopefully those will be released before too long for everybody to use as well. And then we can just look at ADHD symptom scores as a function of these matrices that come from the Gordon or the new probabilistic parcellation. Here's what those look like in ABCD1 and ABCD2. At the top, using all of them, even no matter what you do, there's not a super high relationship, but if you look at the top 1% of the connections, we see a relatively high relationship. We doing these in 2 independent samples of how they're might be correlated. What's really interesting is if you look at the Gordon parcellation, which is down in the bottom, is that no matter at any level, there's a non-significant correlation between, it's essentially non-replicable. So In this case, at least looking at ADHD symptom scores requires a large sample, and you need to reduce the noise with regard to variability and topology across subjects using this probabilistic parcellation. Okay. Now, if we look at our independent sample, which is enriched actually for ADHD, using this Polycon risk score, right? We see a very cool finding where it split up the ADHD and the controls very, very, very widely. You get a relatively large effect size, and you see this decrease as a function of age, which is expected based on changes in symptoms. And so we think we have another angle at which we can do kind of smaller studies, leveraging these larger studies like ABCD and Biobank and things like that. All right, so the end here is that I'm just going to, we're building off the work in genetics, this polyvertex score, Polycon score, or some of these other multivariate methods might allow for increased power to study brain behavior associations in small samples. And importantly, that the combination of these approaches with the regions that account for inter-individual variation will be important to further this goal. So I'm going to end there. I'm just going to, again, say thanks. I'm hopefully that definitely get in contact with me if you want to learn anything more about the ABCC, the community collection. And there's lots of information online on it now. We are definitely in a stage in our field in cognitive neuroscientists where we do have a replication problem, and we need to think outside the box about how we're doing our studies to make sure that we're having real findings. There are some things we can do about it, and I highlighted a few things that we're working on, and I'm hoping that the message that I'm portraying here will lead others to kind of move and think outside the box in the same direction. Again, thank you all for having me. Thanks the enormous lab and collaborators who've helped me conduct a lot of the work that we're talking about here. And of course, thanks to all the different funding that we received to conduct the work as well. I'll end there, and again, just say thank you all for having me, and I'll take some questions.
All right, I muted everyone in case people would like to clap for this excellent talk. All right, so I'm going to mute everyone back. And then Damien, if you can unmute yourself, I'm going to start with some questions. I'm going to just read it off. There's only 2 so far online, but please feel free to add. If we're done with it, you're welcome to chime in and just unmute yourself and talk if we're done. And if Damien can stay on, I know we have until 5:00 p.m., but we'll see. We'll try to finish several minutes early. Okay, so a question from Victoria. Is correlation matrices of each subject are available for download? I show only the correlations between Gordon's networks, but not detailed to ROIs as you demonstrated in the presentation.
Yes, that's a great question. Yes, the correlation matrices kind of using our, my standard, our standards of how to clean them up and do all those are available in the ABCC for people to just download those if you just want to work with them. There's also a tool that allows you to utilize the parcellated time series to generate your own if you want to change the parameters, good data, bad data, things like that, that's all available. The new parcellations are not quite up there yet. They're being uploaded as we speak. And we're going to make them, essentially there's going to be a website where you can download and you can make them yourself however you want. That is being put together. That is being put together right now. The paper is hopefully be finalized before too long. And as soon as we put it up, because this will be, it's going to be more of a resource for everybody. So that will go out in bioRxiv probably before too long, and then everything will be available for people. So they're almost out, not quite.
Great, thank you. Question from Tiffany. When looking at figure 2 of Scott and Brendan's paper, it seems that type 2 errors rather than type 1 are more of an issue with smaller sample sizes. If there is time, I wonder if you could comment a bit on this and how to reconcile that with some of the other points on reproducibility and sampling variability you nicely summarized. Thank you.
Yeah, that's correct. Although you get both, right? Because in essence, you know, you get type 1 and type 2 errors. In essence, because this is kind of hard to, it's always hard to explain. If anybody followed this when the paper was put on a bioarchive, it was just a chatter fest on Twitter of trying to, you know, walk through a lot of the issues. And that is that if the true effect is actually truly really small, then, and you have a small sample size, right? And then you're actually doing the statistics properly, like correcting for multiple comparisons and doing all that thing right, you in essence the true effect will be, you won't see it. It will not pass significance. And that is partially shown, I believe, in that figure. But the real scary part, right, is that you get the other effect too, where if it's just by chance, right, you pull the sample that has a high relationship with what you're looking at, that will pass the multiple comparisons correction, and that will be an inflated effect, even though it's not real. So you get both. The problem in our literature, typically unfortunately, what we see is that we see mostly the latter, because it's the positive effect, and that's easy to get published, even though it's likely to be wrong. So hopefully that helps with that question.
Great, thank you. Other people, is there other questions? Sure, I'll let Damien go. All right, well, congratulations again, Damien, and thanks very much for the tour of the force talk. This is fabulous, and congratulations again.
Yes, thank you very much. Again, thank you all for having me. It's much appreciated.
Thank you. Bye.
Thank you.